One thing I'm really excited about right now is the Sioux Bioreactor. Has anybody heard of it? Google it. It's amazing. It's this, the Sioux Bioreactor. So Johnson spelled like you think it would be spelled -SU. And it's a microbiologist and his wife who developed a low-tech mobile composting system originally for dairy manure, so really highly nitrogenous. It's a wood pallet with holes drilled into it, and then it uses perforated drainage pipe vertically placed onto the pallet. And then he puts concrete reinforcement wire in a circle around it. I think it's about 13, maybe, just think the width of a pallet, right? So it's like a 13-foot panel of that concrete reinforcement wire. You could probably also use like a cattle panel or something. And it's bent around, sitting on top of the pallet. And then you put the drainage pipe perforated, maybe 5 of them, down into the pallet. And then he's welded a rebar ring for the top so that the pipes can be lashed to the rebar ring and they won't move. And then he fills it with material. And the two beautiful things about his system is you can use any material. You can use all one type of material. So in his YouTube video, he uses all wood chips, or it's leaves. And he has them in a wheelbarrow and he puts them in a water bath to get the medium really nice and wet. And it has to be chopped up pretty fine. But then he fills the bioreactor. There's landscape fabric around the reinforcement wire, and then there's landscape fabric over top for shade, for cover. In order to get like the specs for composting safety, it has to be contained. And you have to hit a certain temperature within 24 hours. And so he does reach that PFRP temperature. I think it's above 130 and less than 160 within 24 hours. And then he removes the pipes after 24 hours. And the mycelium of the fungus in 24 hours is already so advanced that it will hold the holes that the pipes had created, even if you take the pipes out. It's awesome. He's got an irrigation, like a 3/4 inch or 1 inch poly pipe ring with sprinkler emitters coming out of it. And he waters it once a day for 1 minute. And he composts for, I think he said it's ready in as early as 3 months, but he leaves it as long as he can. Once it hits that temperature, it'll start to cool and you keep it watered, then it just ages basically. But he said that he's seen microorganisms in there that people haven't seen for like 100+ years, which is really exciting. He did a PowerPoint presentation at the soil carbon thing in Australia, soil carbon summit in Australia. And if you're into this kind of stuff, watch it, because not only does he go over the design again that's in his YouTube video, but he also just talks about what he's seen and how he uses the compost, because depending on the material he uses to compost, he gets a different consistency of compost. And some of the stuff he was showing was like this really sticky, like it was like a picture of his hand just like ribboning the compost up. And that stuff he's using to coat seeds or pumping it through his irrigation, using it for extracts. And he was talking to like big conventional farmers in Australia about using this to like tumble their seeds in this compost sludge that he's making and then drilling them in. And he talks about like the effects of glyphosate on the life in the soil. And I mean, it's a great 45 minutes of your time. His name is somebody Johnson, and he's out of the University of New Mexico. I'd really like to bring him to Living Web next year. Yeah. But I mean, consider that in Santa Fe, where he is, or close to Santa Fe, or he's actually in more close to Taos, so like pretty crazy elevation. He gets really cold temperatures and really hot temperatures, and the compost is fine. The beauty of it is he uses any material he wants and he never turns the pile. And that's why he gets such rich life in it, because he's not disturbing it. And he's able to mature the system, right? So the longer you leave it, the higher successive ecosystem that you'll see. I mean, eventually it'll run out of food, right? So you've got to either feed it or you've got to use it. But another thing he does is once the temperature's dropped, is he'll put in worms. And then that just like takes to this whole other level. But even if you don't add the worms, it's like really, really amazing compost, apparently.
Don't those really high temperatures kill the mycorrhizae? How do they form?
If you keep it between 130 and 160, you'll kill pathogens without killing the beneficial life. So that's that like sweet spot. Okay, where are we? Does anybody else have a sample that we want to pull from a different sample? Okay, where's that from?
10 years I've been working on the soil in my garden.
Like raised bed or?
No, it's not raised bed.
Cool. Does anybody want to make this sample? All right, Kelly. Since you're closer to it anyway.
It's my dirt.
That's your dirt. Hopefully it's soil, not dirt.
Soil.
So she's looking for 1 milliliter of soil mix. Nice, get it nice and mixed up. I'll get your slide ready.
I see a lot of things that look like little spiders.
Oh, really? Are they moving? Are their legs moving? Are they round, like with little things coming off?
Like just little middle section with
So you could be looking at stellate amoeba, which are pretty beautiful. So do the spider legs go out pretty far, or are they short and stumpy? It sounds like stellate amoeba. I might have a picture of one that I can show you, and you can see if that's what you're seeing. So that's a bacterial feeder.
I haven't seen any pictures.
You see a lot of them?
Yeah.
If you have too many protozoa, they'll decimate your bacteria, right? And so then you don't have readily available nutrients coming from bacterial bodies for your plants. So you've got to find some way to keep that population in check. You either need fungi, right? Or food. Some nematodes will eat whatever will fit in their mouth. Bacteria feeders will eat whatever fits in their mouth. Some of them have larger stomas than others, and they'll eat protozoa. But some of them have small and will only eat bacteria. So it just kind of depends on the species. And when we're doing this type of microscopy, we can't discern species. We're looking at genera of organisms, so we can tell. By morphology, we can tell a lactobacillus from a streptococcus, for example, but I can't tell you what species of lactobacilli it is. Does that make sense? I would have to do DNA sequencing in order to figure that out.
So what's up with wood chips being so important as opposed to leaves or?
It's dense, nutrient-rich lignins.
Yeah, so it's stable. It's the stabler form of carbon, so it provides a slow release carbon food for.
What about the actual cell structure in the wood itself, allowing the structure for it to housing, kind of like your biochar?
Yeah, it's a substrate for sure for the organisms to live on. It's also a food source primarily for fungus after it's been broken down by other organisms. But one of the most amazing facts that I learned recently is that even a fungus, if it has been present in the soil and has been able to build up a really strong body, so strong cell walls, strong hyphae, that's basically strictly carbon, the skeletal body of the hyphae, right? So even if that fungus dies, say that soil goes anaerobic, even briefly, and a bunch of anaerobic bacteria come in and suck all the cytoplasm out of that fungus and it dies, that carbon skeleton of the fungus will stay in the soil for 100 to 200 years. So it's still giving you the structural benefit and it's still sequestering carbon. So to have fungus at some point is so much better than to never have fungus at all, right? That's what that tells me. And so like, for example, here we produce biochar. And so, biochar is touted as this like super carbon-rich soil supplement that provides a substrate for microorganisms, blah, blah, blah. It's really good stuff. But also like the byproduct of producing biochar is wood vinegar. And so we've been using the scope to do all these different looks at, okay, if I spray this grass with a full concentration of wood vinegar, what happens? Well, the grass dies. What happens to the soil? And I was able to see that the fungus in the soil was being attacked by anaerobic microbes and that they were congregating all around the fungus. So I looked at the soil before spraying, I looked at the soil after spraying, and right after we sprayed, all the fungi in the soil was surrounded by bacteria. And the only thing I can assume is that those are anaerobes and they're there sucking out the cytoplasm, releasing acidic glues. And so, great. You have an organic pesticide and it kills your grass. Question is, what's it doing to everything else? And the bigger question is, what do you do after you spray, right? So if I spray that grass and all those bacteria go and they suck that fungus dry, well, then say I go back and I throw in some really awesome aerobic medium on top of that, and I get going in the right direction again, then it's okay. You know, it's okay. I'm gonna let the good guys win. But if I keep hitting it with the most organic but super acidic anaerobic product that I can, I'm certainly gonna be killing the beneficial life in the soil or keeping it at early successive state. Does that make sense? So this is what I mean when I say practical management applications of this type of information. Do you want this?
Yes, but before I do that, did I screw this up by using this water?
No, because that's well water and I feel fine about it. It's not chlorinated. I'm just gonna put that right there for you. And you're gonna need cover slip. All right, so remember you wanna take the edge of it and just gently spread that droplet out and then drop it. Great, very nice. All right, so here we are at 40x. I'm just going to find a corner. Let's go top left. And just scan. See if you see any big whip-like worms moving around or any crazy-looking monstrous insects. If this is from Dee's garden, we probably won't see microarthropods, but just in case. And I can show you a couple pictures of.
They're pretty fun looking.
They're so awesome. And I have a picture. They're kind of scary.
Yeah, I look at moss and you find stuff in moss all the time.
Oh, that's a great idea. Well, yeah, really, anaerobic soils are much more fun to look at than garden soils, and forest soils are way more fun to look at than. You'll find microarthropods in forest soils way more readily than agricultural soils. And it's a little less common to find testate amoeba in agricultural soils. But I find them often in worm castings and like in your perennial garden, you might find them. There's our bubble.
Those Lancaster berries, by the way, will attract worms to your garden.
So we're scanning through, we're not seeing much. There's a whole lot of stuff in there. So you would know right away with this sample that you need to dilute quite a bit if you wanted to get an accurate count on bacteria. Ooh! That may be a piece of a microarthropod. Or it could be some root hairs. We'll have to go back to that. Well, why don't we go ahead and just take it on down. I found this, it would look like a feather, but it was like skeletal looking in the soil. And I sent it to my mentor. I said, what is this? And she said, it's a microscopic Russian listening device. And then she emailed me back and she said, no, it's an antenna from a microarthropod. I was like, oh. It was funny. Here it is. Cool. So I don't know if you can see it from where you are, but like, you can't. But in the scope, it's like totally dimensional. Like the spikes coming off of it, like some of them are coming towards me and some of them are facing down. Let's zoom in on it again. Let's see what we see. So at 400x, it's like there's an aggregate around it, like the base of it. See the fuzziness around the base of it? So under the scope, I can clearly see that that's just like a bunch of aggregation around it. So I can't really see. I mean, I'm guessing it's some kind of like plant material. Just because I don't see like any segmentation, like it's not legs.
The piece at the top does have some, pull it back down, go to the left, or your right.
I'm sorry. Yeah, I see what you mean. But that, looking at it too, it could also be like a detached cell wall or something, just like looking at the, so fun, these things. What is that? I don't know. Maybe we have crushed a critter here. It almost looks like a leg or something. How exciting. So you can see we kind of have two layers. Like here's a layer of mostly aggregate. And if I focus differently, you get to see like all the things under it. And we're in a very busy area right now, which makes it so hard to see. Let's try to head out. Well, I would dilute it more if I wanted to see what was going on in there, you know, inside of all that. Oh, look at that. See that little guy, little pill looking thing? See how it's dark and segmented? It's likely a fungal spore. And so that's not necessarily like, you can't be like, oh, sweet, I have fungus, because that thing could take 500 years to germinate. I mean, it could take a long time. It could germinate as soon as we pull the slide off of the thing, or you have no idea how long that thing is gonna be, camping out. So you can't really count it. The only thing you would wanna note is, do you see more of them? And does it look like ooh, is that look, it's an amoeba. It's an amoebal cyst. So exciting, look at all this.
The bottom right?
Do you see do you see the internal organs and the cell wall?
And they eat bacteria?
Mm-hmm. And if you looked in there, if you could see it really, really closely, you would probably see bacteria in there. So they're not necessarily eating while they're encysted, but everything just sort of froze. So if it had pulled a plug out of the soil and was eating bacteria when it encysted, that bacteria is probably still in there, maybe slowly digesting, but it's not like actively eating. Let's see what else we can see here. Looks pretty good, Dee. You got some great aggregation. You got good bacteria, protozoa.
These are hugelkultur beds.
Oh, they really.
Yeah.
So were they wood? Was it wood? The base was wood?
Yeah, poplar,
Okay.
mostly poplar, and the turkey mushroom is growing in it.
Oh, good.
And I also bought mycorrhizae stuff,
Did you?
and I wonder if
That's actually probably not going to be very effective right now. I would
let
think.
me see it.
Oh, there's a little fungal hyphae.
It needs roots of plants to trigger,
Oh.
and they have to be really close to the roots. The ectomycorrhizae would work there.
This was a couple years ago when I applied it.
Yeah.
It just depends. So it depends on the source of the mycorrhizal inoculant. If they've chopped up roots to produce it, it's not going to be as effective. But if they've actually isolated extramatrical spores, like that we talked about earlier, through a scope, then that's going to be more effective because you know that you have something that's going to sprout and find a root or something. You'll just have to look at the package or the website of the company where you got it and see if they tell you how they derive the inoculant. So that's a really decent fungal hyphae right there. It's not humongous, but it's wide, larger than, so the way we measure them is we take the smallest bacteria in the field of view, and then you consider that 1 micrometer, and then you add up how many of those bacteria it would take to get across the width of the fungus. And then, you measure the length of the fungal hypha based on the length of the field of view. And so greater than, I believe it's 2.5 micrometers wide is considered beneficial. So, that's definitely greater than 2.5 micrometers. So, and it's dark and the septae are regular from what I can see under the scope. I don't know guys if you guys can see it from where you are.
To me, it's so small that it's just forming. Are those things, they take a long time to grow?
Or it could,
So you don't ever see like a web of them?
you wouldn't necessarily see a web, but you remember that picture I showed you in the PowerPoint with the big fungal hyphae that was like crossing the entire field of view? That was not an old fungal hyphae. So this could be, we broke it. You could have broken it off when you took the soil sample. It could be on the young side, but one thing is that it's harder to tell on darker fungi than on lighter colored fungi, because sometimes in the lighter colored you can see the cytoplasm on the inside and you can see the vacuoles. And you can see, like when a fungus is growing, it shoots its cytoplasm towards the growing tip. So you can tell if something's newer, right? That it has a lot of vacuoles and it has a lot of cytoplasm in it, but we can't really tell from looking at that.
Like 80 pounds of pressure. It's amazing it doesn't blow up.
They're so amazing. They will, if they are like growing their hyphae in one direction and they stop finding food, they can actually back their cytoplasm out and send a branch out from another part of the fungus.
Do that, it's really kind of geeky.
Really?
They would water.
Oh, look at our samples drying. You see that? Whoop. So yeah, you could really waste a day looking at. I mean, if you get really into this. And it's really fun to look at anaerobic organisms. They're so exotic looking.
Tardigrades and moss and stuff too.
Tardigrades are awesome. And like aquatic systems have their own like little world of organisms. Oh, bye. There's some fungus. Do you see those little fungal hyphae all in there? That's good, Dee. Oh yeah, definitely. We saw some spores, didn't we?
I've only tilled this like 2 times in the 10 years. And otherwise I just kind of take a
How
digging
deep
fork
is it?
and loosen it up.
Yeah.
Because I didn't know about tilling, still trying to figure that out.
The more I look at soil under the microscope, the less I want to till ever again. The other thing that, I mean, I was going to go into like management applications later, but we can talk about it now. So if you think about, like, say an extra matrical mycorrhizal spore does end up in your soil or is there, has 48 hours to find a plant root after it germinates. That's really not that much time. So think about, like, even in, highly organic systems, if you have constantly tilled soil or at least annually tilled soil and complete annual crops that are all going to be ripped out, all those roots are going to be ripped out of the soil. And then, yeah, maybe you're planting cover crops, but how much time are you actually giving mycorrhizal to establish and to thrive, right? Because every time you have a great endomycorrhizal colonization of your onion crop, you go in there, you harvest all your onions, you disturb all the hyphae. And if there's no perennial there to support that network, it's just going to die. And you're going to have to reestablish it, right? So this is a huge argument for intercropping perennial and annual species. This is the argument for silvopasture. This is the argument for permaculture, like no-till, all that stuff, So it's not a matter of convenience for me anymore whether I put perennials in my annual beds, I have to put perennials in my annual beds.
Mycorrhizal stuff is even more tenuous too, because some of them, the glomeromycetins can only sense. They have to be like within a quarter inch or something crazy.
Really? Of where the exudate is? Mm-hmm. Yeah, sensitive little stuff, And as much as we're in there messing around, you can imagine how many attempts we're dashing, You guys want to take a break for a little bit and then get on the scopes?